flower through human eyes, and you see solid colors designed to attract attention. But if a bee shifts the spectrum, that same flower reveals a hidden map of bright, ultraviolet landing strips pointing directly to the nectar. We often assume our single-lens eyes represent the evolutionary pinnacle of sight. Insects had to move beyond the passive reception of light. They evolved to treat photons as a survival tool. This schematic outlines the basic biological hardware to use light: a lens to focus incoming rays, a receptor to catch them, and a nervous system for translation. In insects, this receptor is the rhabdom, a dense matrix of microvilli housing visual pigments that physically catch falling photons. By adjusting the structure of this core rhabdom, insects have developed a sliding scale of optical technology to master their specific environments. At the simplest extreme of insect sight, species like aphids utilize dermal detection, sensing light directly through receptors embedded beneath their body surface. This line graph illustrates how aphids use this unseen hardware. They are not seeing shapes, they are measuring time. As the photoperiod crosses a critical threshold, it triggers an internal biological clock that dictates their seasonal mode of reproduction. Moving up the scale, the simplest actual lenses belong to larval insects like caterpillars. These are called stemmata. Stemmata provide very low resolution, so caterpillars compensate with physical movement. By scanning their heads from side to side, they stitch together a functional view of their surroundings from individual points of light. Adult flying insects often possess a specialized version of these simple eyes, called ocelli. Typically arranged in a triangle on top of the head, they integrate light over a massive visual field. Instead of forming a sharp picture, the ocellar lens intentionally blurs the world. This allows the insect to bypass complex image processing and use the ocelli as high-speed horizon detectors, instantly correcting pitch and roll mid-flight. At this level of biological design, evolution prioritizes processing speed and raw environmental data over high-definition imagery. For complex vision, insects rely on the compound eye, which functions as a kaleidoscopic array of thousands of individual visual units called ommatidia. In optics, there is a constant physical trade-off. A lens system cannot provide both high-definition resolution and maximum low-light sensitivity at the same time. This cross-section shows the apposition eye. Insects isolate each ommatidium by dropping dark pigment walls between tubes, ensuring light travels straight down a segregated channel, producing a sharp mosaic for hunting in sunlight. Nocturnal insects use an optical superposition eye, retracting pigment walls to create a wide, clear zone. This allows light from multiple adjacent lenses to overlap onto a single receptor. It sacrifices image sharpness, but significantly increases the volume of photons captured for survival in near darkness. To capture even more light, many nocturnal insects add a tapetum, a mirror-like layer at the base of the eye that bounces mist light back through the cells for a second chance at absorption. This biological mirror is exactly what causes the eerie, glowing eye shine when a flashlight beam straights a moth in complete darkness. The rhabdom also offers a navigational superpower. Its microvilli are inherently sensitive to the plane of polarized light. This gives many insects a built-in sky compass, allowing them to calculate the sun's position even when it's hidden behind clouds. The compound eye is a highly customizable optical platform, its specific design dictated entirely by the physics of the insect's environment. Up until now, we've looked at how insects capture photons, but some have evolved the biological machinery to actively generate them. In specialized beetles and flies, the insect shifts from a passive observer to an active source of light. This diagram illustrates the chemical factory inside a firefly. The insect mixes luciferase with oxygen and ATP, a cellular energy source. This chemical reaction emits light. By varying the release of ATP, the insect dictates the timing, duration, and rhythm of flashes, creating a visible language. Males and females use these species-specific flash patterns to identify each other, signaling back and forth to coordinate courtship in the dark. This reliance on specific optical signals creates a vulnerability that some predators exploit. The female Photorus firefly mimics the mating signals of other species, By broadcasting the correct visual rhythm, she lures in unsuspecting males for cannibalization. The most extreme application of this technology belongs to the Arachnocampa fungus gnats, which live in the absolute dark zones of cave systems in Australia and New Zealand. These gnats illuminate sticky threads suspended from the ceiling. By mimicking a starry night sky, the glowing traps lure prey upward into the threads in a pitch-black environment. Insects use light to measure time, stabilize flight, navigate, and hunt. They operate a suite of advanced optical technologies that allow them to manipulate a dimension of reality we are only just beginning to understand.